This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome. Today, we explore a fascinating genetic paradox. Why do the mismatched X and Y chromosomes still share a specific set of genes? We'll travel back 300 million years to their origins as identical twins and trace the evolutionary battle to preserve their shared molecular handshake. You'll discover how these surviving genes orchestrate our biology, from bone growth to immune response, and why this ancient partnership is essential for human health. Join us as we uncover the hidden bridge between our most different chromosomes. Chapter 1: The Identical Twins of Our Genetic Past. If you were to examine a human karyotype treated with G-banding, the specific staining technique used to reveal the unique dark and light patterns of our genetic material, your eyes would likely be drawn to the 23rd pair of chromosomes. In a male, this pair appears as a striking biological mismatch. One member, the X chromosome, is a large, robust structure packed with thousands of genes. Its partner, the Y chromosome, is a tiny shrunken vestige. Carrying only between 45 and 100 genes, the Y chromosome has the lowest gene count of all 46 human chromosomes. For decades, these two were viewed as biological opposites, one expansive and one diminished. However, the genetic code reveals that this mismatched appearance is a relatively recent development in the grand timeline of life. To understand why certain genes still exist on both the X and Y chromosomes today, we must travel back roughly 300 million years to a time when they did not exist as sex chromosomes at all. At that point in our evolutionary history, the X and Y were a perfectly matched pair of ordinary autosomes, indistinguishable in size, shape, and function. They were identical partners that exchanged genetic material freely, just as our other 22 pairs of chromosomes do today. The transformation of these identical twins into the mismatched pair we see today began when one member of that ancestral autosomal pair acquired a specific mutation, a genetic master switch that could trigger the development of a male. In the lineage that led to mammals, this switch was the SRY gene. Once this gene appeared on one chromosome, that chromosome became the proto-Y, while its partner became the proto-X. This master switch initiates male development by triggering the secretion of anti-Müllerian hormone to regress female internal structures and testosterone to develop male accessory structures like the epididymis and vasa deferens. Initially, the two chromosomes remained nearly identical, but the presence of a sex-determining factor set off a chain reaction that would eventually strip the Y chromosome of nearly all its original identity. The primary driver of this separation was the suppression of recombination. In a normal pair of chromosomes, recombination acts as a vital repair and mixing mechanism. During meiosis, homologous chromosomes pair up and swap segments of DNA, ensuring that harmful mutations are purged and beneficial combinations of genes can spread through a population. However, once a sex-determining gene like SRY is established, natural selection begins to favor the cessation of this genetic swap. This is because certain genes are better for one sex than the other, a phenomenon known as sexually antagonistic alleles. A gene that boosts male fertility but is detrimental to female health is an evolutionary advantage if it remains on the Y chromosome, near the SRY switch, where it is always inherited by males. If recombination were to occur, that male benefit gene could be swapped over to the X chromosome and end up in a female, where it would cause harm. To prevent this, the genome utilizes inversions, segments of the chromosome that flip upside down, making it impossible for that region to align and swap DNA with its partner. This suppression of recombination did not happen all at once across the entire chromosome. Instead, it occurred in discrete chronological steps that scientists can still map today as evolutionary strata. In the human genome, we can identify 5 of these distinct strata on the X chromosome, labeled S1 through S5. Each stratum represents a specific moment in time when a new inversion event occurred, locking a fresh segment of the chromosome away from its partner. These strata serve as a physical fossil record of the Y chromosome's decline. The oldest layer, S1, dates back to the very beginning of the mammalian line over 300 million years ago. The layers then progress in age until reaching S5, the youngest stratum, which formed only about 30 million years ago. A fascinating detail of this physical layering is that S5 is located immediately adjacent to the pseudo-autosomal region at the tip of the chromosome. This means the history of the chromosome is literally layered from the oldest events at one end to the most recent events at the other. Each time a new stratum formed, more genes were trapped on the Y chromosome in a zone where they could no longer be repaired by swapping DNA with the X. Cut off from the restorative power of recombination, the Y chromosome entered a fundamentally different evolutionary environment. Without the ability to purge harmful mutations by swapping them for healthy sequences from the X, the Y became subject to a process known as Muller's ratchet. In this scenario, the least mutated version of a non-recombining chromosome can be lost from a population by sheer chance. Once that perfect version is gone, the ratchet clicks forward, and the overall quality of the Y chromosome drops. It cannot go backward. It can only accumulate more damage. Over millions of years, this lack of repair led to a massive loss of genes. The Y was hit by a barrage of deletions and mutations that rendered its original genes non-functional. While the X chromosome remained relatively stable and gene-rich because it still recombines with another X in females, the human Y chromosome lost about 97% of its original autosomal genes. The shrunken state of the Y is not just a story of loss, but also one of specialized survival. As the original autosomal genes decayed, the Y chromosome developed unique structural features to protect its remaining functions. Among these are long inverted repeats called palindromes. These sequences allow the Y chromosome to engage in a form of self-healing called gene conversion. In this process, the chromosome copies a functional version of a gene from one side of a palindrome to a damaged version on the other. It is a lonely form of recombination that does not require a partner, but it is one of the few ways the Y can maintain its remaining vital functions in the face of relentless mutation. This evolutionary trajectory is not unique to mammals. Similar patterns occur independently across the tree of life. In birds, the system is reversed. Females carry the mismatched ZW pair, where the W is the shrunken, non-recombining partner. We even see this in plants, such as walnuts and hickories, where genomic regions controlling flowering order show the same signatures of sex chromosome-like evolution, including the accumulation of repetitive DNA and a lack of recombination. It appears that whenever a chromosome pair is forced to stop swapping information, one of them inevitably begins to degrade. One of the most important living fossils in this story is the platypus. As a monotreme, the platypus occupies a unique middle ground between reptiles and therian mammals, having diverged from the lineage of therian mammals approximately 165 million years ago. Its sex chromosome system is incredibly complex, consisting of 10 chromosomes that form a chain during male meiosis. What makes this system so important for understanding our own history is that it contains genes related to both the bird ZW system and the mammal XY system. This suggests that the ancestors of all mammals may have once utilized a system that was much more bird-like. While the origin of the mammalian XY system is ancient, dating back roughly 300 million years, the platypus shows us that the refinement of the specific boundaries and the stabilization of the system we see in humans today occurred more recently than once thought. The platypus provides a snapshot of a time when the boundaries between these different sex-determining systems were still fluid, bridging the ancient autosomal past and the specialized chromosomal future. Despite the massive divergence and the loss of thousands of genes, the X and Y chromosomes have not become entirely different entities. They still share a small set of genes that have resisted the pull of decay, such as the influential regulatory pair ZFX and ZFY. These survivors have remained functional on both chromosomes because their roles in managing the expression of thousands of other genes are too critical to be lost. Furthermore, the chromosomes maintain a physical connection through the pseudo-autosomal regions, or pars. These segments at the tips of the X and Y still look and behave like the original autosomes, allowing the two chromosomes to pair up during the production of sperm. These shared regions and genes are the threads that continue to bind the X and Y to their identical past, serving as the essential anchors for both the mechanics of inheritance and the regulation of the entire human genome. This ancient identity is what allows the two most different chromosomes in our body to find each other in the dark and begin the intricate dance of meiosis. Chapter 2. The Molecular Handshake. Docking the Mismatched Pair. To understand why the X and Y chromosomes still share a specific set of genes, we must look at the most critical moment in the life cycle of a cell, the process of meiosis. This specialized form of cell division produces sperm and eggs, and it is here that the mismatched nature of the sex chromosomes presents a massive logistical challenge. In every other pair of chromosomes in the human body, the process is straightforward. Chromosome 1 finds its identical partner, chromosome 2 finds its partner, and so on, all the way down to 22. They align perfectly, zip together, and exchange genetic information through recombination. But the 23rd pair is different. In a male, you have the massive gene-rich X and the tiny shrunken Y. They are heteromorphic, meaning they are morphologically dissimilar. They do not look alike, they do not carry the same genes for most of their length, and they cannot align across their entire span. Yet, for meiosis to work, they must find each other, pair up, and segregate into different cells with absolute precision. The biological solution to this docking problem lies in the pseudo-autosomal regions, or pars. These regions are the remaining bridge between the X and Y. They are called pseudo-autosomal because the genes within them do not follow the typical rules of sex-linked inheritance. Instead, they behave exactly like genes on the autosomes, the non-sex chromosomes. In an autosome, you inherit one copy from your mother and one from your father, and those genes can be shuffled and swapped in every generation. The same is true for the genes in the pars. Even though they are physically sitting on the sex chromosomes, they exist in two copies in both males and females. Because these regions are nearly identical on both the X and the Y, they are capable of the same molecular handshake that the autosomes perform. They allow the mismatched X and Y to recognize each other as partners. Think of them as genetic docking stations located at the very tips of the chromosomes. In humans, we have two of these regions. Par 1, the larger segment at the tips of the short arms, and Par 2, a much smaller segment at the tips of the long arms. PAR1 is the heavy lifter of the two. Spanning about 2.7 million base pairs, it is the primary site where the X and Y chromosomes align during the first phase of meiosis, known as prophase 1. This alignment is not just a passive meeting, it is an active, mandatory event. For the sex chromosomes to separate correctly and move to opposite ends of the dividing cell, they must undergo what biologists call an obligatory crossover. This means that in every single instance of male meiosis, at least one exchange of genetic material must occur within the PAR1 region. This crossover creates a physical link between the X and Y, holding them together so the cell can pull them apart correctly, ensuring one sperm gets an X and the other gets a Y. If this single crossover fails, the chromosomes can drift apart prematurely, leading to a total arrest of sperm production or the creation of gametes with the wrong number of chromosomes. Because this tiny region of DNA is responsible for the pairing of the entire 23rd pair, the molecular activity within it is staggering. The recombination rate in PAR1 is roughly 10 to 17 times higher than the average rate found across the rest of the genome. It is a massive hotspot of genetic shuffling. Within this high-octane environment, PAR1 houses 24 distinct genes. These are not just structural anchors, they are functional genes that play diverse roles in human biology. For example, the gene CD99 is involved in cell adhesion and the migration of white blood cells, while PLCXD1 plays a role in cell signaling. These shared genes, such as shocks, are so vital that having too few or too many copies leads to the physical traits seen in conditions like Turner or Klinefelter syndrome.
The mechanics of how these regions find each other are a marvel of molecular engineering. During the early stages of meiosis, the ends of the chromosomes, the telomeres, attach to the inner membrane of the cell's nucleus. They move along this membrane, searching for their homologous partners. For the X and Y, the pseudo-autosomal regions are the only places where the DNA sequences match well enough to allow the proteins of the synaptonemal complex to zip the two chromosomes together. This complex is a ladder-like structure composed of specialized proteins that hold the chromosomes in perfect alignment. In males, this zipping process is restricted to the PARs, creating a physical bridge that allows the recombination machinery to initiate a crossover. As the synaptonemal complex forms, the sex chromosomes are sequestered into a specialized territory within the nucleus called the XY body, or sex body. This is a dense mass of chromatin that can be clearly seen under a microscope. The formation of the XY body is a protective measure. It keeps the mismatched sex chromosomes separate from the rest of the genome during the middle stages of meiosis. While the rest of the X and Y chromosomes are largely silenced within this compartment to prevent interference with the cell's division, the pseudo-autosomal regions remain active and accessible. This allows the handshake to be completed even while the sex-specific regions are locked away.
One of the most unique features of the PAR is its relationship with the XG blood group system. The XG blood group is a classic anecdote in genetics because it is the only blood group system in humans located on a sex chromosome. The gene responsible for this blood group is situated right at the boundary of the PAR on the X chromosome. Because of its location, it serves as a vital molecular marker for researchers studying how the boundary between shared and sex-specific DNA is maintained. Another fascinating biological fact tied to these shared regions is the synthesis of melatonin, the hormone that regulates our sleep-wake cycles. The gene ASMT, which is essential for melatonin production, is located within the PAR. This means that the very mechanism that helps us sleep is rooted in the same genetic territory that allows our sex chromosomes to pair up. While PAR1 handles the mandatory pairing, PAR2, located at the other end of the chromosomes, is much smaller and less active. Spanning only about 330,000 base pairs, it contains only 5 genes. Unlike the obligatory crossover in PAR1, recombination in PAR2 is facultative, meaning it only happens in about 1% of male meioses. Among the 5 genes in this region are VAMP7, which is involved in the trafficking of vesicles within cells, and IL9R, a receptor for an important immune system signaling molecule. Because it recombines so infrequently, PAR2 is in a different evolutionary state than PAR1. The gene pool within it is not as homogenized between the X and the Y, allowing the 2 versions of these 5 genes to slowly drift apart.
The genes within these regions are also unique because they escape X inactivation. In typical female cells, one of the 2 X chromosomes is silenced to prevent a double dose of gene products. However, because genes in the PARs are present on both the X and the Y in males, females need both of their X-linked PAR genes to remain active to maintain a balanced dosage between the sexes. This ensures that whether a person is XX or XY, they have 2 functional copies of genes like CD99, PLCXD1, and ASMT. This requirement for balance highlights that the pseudo-autosomal regions are not just mechanical docking stations, they are essential components of the body's broader genetic equilibrium. The high density of genes in the pseudo-autosomal regions is another point of interest. These areas are actually more gene-dense than the rest of the sex chromosomes. Because these genes are constantly being recombined in males, they have a different evolutionary signature than the genes in the non-recombining sex-specific regions. Recombination allows for more efficient natural selection, which means these genes can adapt and change without being dragged down by the accumulation of harmful mutations that plagues the rest of the Y chromosome. The pars act as a refuge, a small territory where the Y chromosome can still participate in the restorative process of genetic exchange.
This molecular partnership is the foundation upon which the rest of our sex-linked biology is built. It is the silent, high-stakes maintenance work that allows the specialized functions of the X and Y to exist without collapsing the entire system of inheritance. The shared genes in these regions are the evidence of this partnership, serving as both the anchors for meiosis and the contributors to vital processes like immune response and sleep regulation. But the influence of these shared regions extends far beyond the mechanics of cell division. If these genes are so essential that they must be preserved on both chromosomes to ensure proper pairing and dosage, what does that tell us about their role in the rest of the body? The physical connection made during meiosis is only the beginning of a much deeper regulatory relationship that spans the entire human genome.
Chapter 3: The Genomic Conductors. How Gametologists Orchestrate the Sexome. When we think about the X and Y chromosomes, we usually think of them as the master architects of biological sex. We imagine them as specialized blueprints that contain the instructions for building a male or a female body, and once that job is done, we assume they simply fade into the background. However, recent scientific research has revealed that this view is far too narrow. The sex chromosomes are not just the designers of our reproductive systems. They are influential, lifelong regulators of a massive genomic network that spans the entire body. While they certainly determine whether an embryo develops testes or ovaries, their most widespread impact happens far away from the reproductive tract, in the heart of every single cell. This realization has shifted our understanding of the sexome, the total network of genes across all 23 pairs of chromosomes that are dialed up or down by the presence of X and Y genes. At the center of this regulatory network is a suite of genes known as gametologues. These are homologous gene pairs that have been preserved on both the X and Y chromosomes despite hundreds of millions of years of evolutionary divergence. While the Y chromosome has lost the vast majority of its original autosomal genes, retaining only about 45 to 100, it has fiercely protected these specific pairs. The most prominent among them are ZFX on the X chromosome and ZFY on the Y. To understand how these two genes exert such a massive influence, we have to look at what they actually do inside the nucleus. ZFX and ZFY are transcription factors. In the world of genetics, if a gene is a piece of sheet music, a transcription factor is a manager who decides how loudly each instrument should play. These proteins do not build structural parts of the body themselves. Instead, they travel through the nucleus and bind to specific regions of DNA on other chromosomes, the autosomes. Once they bind to these sites, they act like molecular volume knobs, either increasing or decreasing the activity of thousands of other genes.
For a long time, these shared gene pairs were viewed as evolutionary leftovers, remnants of the ancient autosomal past that hadn't quite disappeared. But research led by David Page and Adriana San Ramon at the Whitehead Institute has shown that they are anything but redundant. By utilizing the natural variation of sex chromosomes found in the human population, these researchers mathematically modeled exactly how much influence these genes have.
They looked at tissue samples from individuals born with atypical chromosome counts, such as those with Turner syndrome, XO, or Klinefelter syndrome, XXY, as well as rarer cases of people with 3, 4, or even 5 X or Y chromosomes. By measuring the levels of gene expression across the entire genome, they discovered a striking pattern. The impact of the sex chromosomes is linear. For every additional X or Y chromosome present in a cell, the expression of thousands of other genes changes by a predictable, consistent amount. Each extra chromosome is like another click on the volume knob, adding more of these global regulators to the mix. To ensure these findings were universal, the researchers focused on two very different types of cells, lymphoblastoid cells and fibroblasts. These were chosen primarily for their ease of acquisition. Lymphoblastoid cells are derived from blood, and fibroblasts from skin, allowing for a broad sample size. What they found was that while the sex chromosomes influenced thousands of genes in both cell types, the specific genes affected were different in each. This suggests that the gametologues are reading different instructions depending on the tissue they inhabit. In an immune cell, they might dial up genes related to pathogen defense, while in a skin cell, they might focus on the structural integrity of the tissue. This tissue-specific regulation helps explain why biological sex can influence the progression of diseases that seem to have nothing to do with reproduction, from heart disease to autoimmune conditions. One of the most surprising discoveries in this research is the functional equivalence of ZFX and ZFY. Despite 300 million years of separation, these two genes have remained almost identical in their regulatory effects. When scientists suppressed their activity in a lab setting, the coordinated changes in gene expression across the rest of the genome largely vanished. This tells us that this single pair is responsible for about half of the total regulatory influence that the sex chromosomes exert on the rest of the body. They have stayed the same because their role is too critical to change. If they were to drift apart, the delicate balance of gene expression required for survival might collapse.
ZFX and ZFY are not the only conductors in this shared regulatory network. Other gametologues provide concrete examples of how shared genes impact development far beyond sex organs. For instance, the gene pair NLGN4X and NLGN4Y is essential for neurological development. Mutations in these genes have been associated with autism and intellectual disabilities, showing that these XY pairs are vital for the wiring of the brain. Another pair, DDX3X and DDX3Y, is involved in RNA metabolism and protein synthesis, serving as a fundamental housekeeping system for the cell. While these genes provide the basic mechanics of being a functioning human, recent research indicates that subtle functional differences between the X and Y versions of this pair contribute to sex-biased biology and disease susceptibility. Even in these essential roles, the slight divergence between the two chromosomes can create distinct biological signatures in males and females. To distinguish between the effects of these chromosomes and the effects of sex hormones like estrogen and testosterone, researchers utilize the 4-core genotypes mouse model. This model allows scientists to decouple the SRY gene from the Y chromosome. By moving SRY to an autosome, they can create 4 types of mice: XX with ovaries, XX with testes, XY with ovaries, and XY with testes. This elegant tool has proven that many biological differences between the sexes are driven directly by the presence of XX or XY chromosomes themselves, independent of the hormones produced by the gonads. It confirms that the gametologues are exerting a foundational influence that exists before and alongside the influence of hormones.
This shared regulatory landscape also forces us to rethink the nature of the X chromosome. In females, one X chromosome is typically silenced to prevent a double dose of gene products. However, we now know that this silencing is not total. A significant number of genes on the inactive X actually escape this process and remain active. ZFX is one of those escapees. This means that in a typical female cell, there is an active X chromosome and an inactive X that is still pumping out these global regulators. In a male cell, you have the active X and the Y chromosome, which is also pumping out its version, ZFY. From a regulatory perspective, the inactive X and the Y are two sides of the same coin. They both function as specialized managers that balance the output of the genome. The active X is the universal partner that everyone has, while the inactive X and the Y are the sex-specific managers that fine-tune the system. Another important player in this network is ZBED1, located in the pseudo-autosomal region. Like ZFX, ZBED1 is present on both the X and the Y and functions as a transcription factor. It is particularly active in the nucleus, where it binds to DNA elements related to cell division and protein metabolism. One of its primary targets is the gene for histone H1, a protein that helps package DNA. By regulating how cells divide and grow, ZBED1 influences the structural foundations of our tissues. The importance of this regulation is underscored by its association with Sotos syndrome, a rare genetic condition characterized by significant overgrowth in childhood, demonstrating what happens when the control of cell division is disrupted. Because it is located in the region where the X and Y still recombine, it is kept in a state of high sequence identity, ensuring that both males and females have two functional copies of this vital growth regulator. The impact of these shared regulators is particularly evident when we look at sex chromosome aneuploidy. In conditions where a second sex chromosome is missing, thousands of genes throughout the body are underexpressed because the regulatory strength is halved. Conversely, in individuals with an extra X or Y chromosome, the overexpression of these global regulators can push the genomic balance out of its ideal range. These are not just male or female symptoms, they are the systemic consequences of a regulatory system that has been pushed out of equilibrium. This network of influence also sheds light on why males and females often have different risks for certain diseases. If the gametologues dial up certain immune-related genes more strongly in one sex, it creates a biological baseline that makes an autoimmune response or a specific cancer risk more likely. The sex chromosomes are not just determining which organs we have, they are setting the thermostat for how our cells respond to the world. The evolutionary persistence of these shared regulators is a testament to the fact that our genome is a highly integrated system. The X and Y chromosomes may look like they are living very different lives, but through genes like ZFX, ZFY, NLGN4, and DDX3, they are still working together to manage the same vast landscape of autosomal genes. This shared role is likely the primary reason why the Y chromosome has not completely disappeared. While it has lost most of its structural genes, it has held on to the regulators that are essential for the viability of the entire organism. If the Y were to lose its regulatory conductors, the resulting dosage imbalance in males would be catastrophic.
This framework also explains why the effects of sex chromosomes can be so subtle and yet so pervasive. When researchers look at how an extra X or Y chromosome affects a specific gene on an autosome, the change in expression might only be 10 or 20%. In isolation, that might seem small. But when you multiply that change across thousands of genes, the cumulative effect on the cell's physiology is massive. It is the difference between a perfectly tuned engine and one where every single part is just slightly out of alignment. Over a lifetime, these subtle shifts in the genomic volume add up to the significant biological differences we see between individuals. As we continue to map these interactions, we are finding that the conductors are even more active than we previously thought. They don't just bind to a single site, they often create complex feedback loops and interact with other proteins to modify the very structure of chromatin. This means ZFX and ZFY can influence whether a particular region of the genome is tightly packed and silent or open and active. They are not just turning the volume knob, they are reorganizing the entire stage. This deep level of integration means that any change in the sex chromosomes has ripples that travel through every biological pathway, from how we metabolize drugs to how our brains process social information. By reaching into the gene networks of every other chromosome, they ensure that the biological identity of the organism is consistent from head to toe. This global influence is what makes the study of gametologues so vital for the future of personalized medicine, as it reveals the ancient, shared conductors that have been leading the performance of the human genome for hundreds of millions of years. This ongoing partnership between the X and Y chromosomes is what allows them to manage the vast complexities of the human genome, a task that requires a level of coordination that extends far beyond the simple determination of sex, a coordination that becomes most visible when we examine the precise mathematical balance of gene dosage.
Chapter 4: The Price of Imbalance, Dosage and Human Health. In the intricate world of genetics, there is a fundamental principle that governs almost every aspect of our development, the principle of dosage. For the vast majority of our 23,000 genes, the human body is calibrated to function with exactly 2 copies, one inherited from the mother and one from the father. This rule of 2 ensures that the proteins produced by these genes are created in just the right amounts to build healthy tissues, regulate complex hormones, and maintain a stable internal environment. When this balance is tipped, even slightly, the biological consequences can be profound. Nowhere is this more evident than in the study of our sex chromosomes. Because the X and Y are so mismatched, they present a unique challenge to the body's dosage requirements. While the rest of our genome relies on the symmetry of autosomal pairs, the sex chromosomes have had to evolve specialized workarounds to manage the fact that males have one X and one Y, while females have 2 X's. The genes that still exist on both chromosomes are the key to this balance, and when their dosage is altered, it ripples through every system in the human body. To understand why gene dosage is so critical, we can think of the genome as a highly sensitive biological thermostat. If the thermostat is set to 2, the organism thrives in a state of equilibrium. However, if the dosage drops to 1 or rises to 3 for certain essential genes, the entire system begins to struggle. This is why sex chromosome aneuploidy, the condition of having an atypical number of X or Y chromosomes, provides such a powerful window into the function of shared XY genes. These conditions are not just about reproductive differences. They are systemic biological shifts that affect everything from the length of our bones to the way our brains process language. The common denominator in these conditions is the dosage of genes located in the pseudo-autosomal regions and those that escape X inactivation. Because these specific genes are active on both the X and the Y, they are the only parts of the sex chromosomes where the body truly expects a dosage of 2 in both sexes. The most striking example of this dosage effect is found in the SHOX gene, located in the primary pseudo-autosomal region, PAR1. The SHOX gene is a master regulator of bone growth, specifically responsible for the development of the long bones in our arms and legs. In a typical male or female, there are 2 active copies of SHOX, one on each X in a female, and one on the X and one on the Y in a male. This balanced dosage produces an average height. However, when we look at Turner syndrome, where an individual is born with only one X chromosome and no second sex chromosome, the dosage of SHOX drops to 1. This single-copy state is the primary reason why women with Turner syndrome are significantly shorter than average. The clinical impact of this specific deficiency is often diagnosed as Léri-Weill dyschondrosteosis, a skeletal disorder characterized by shortened forearms and lower legs. A hallmark of this condition is Madelung's deformity, a painful misalignment of the wrist where the radius bone grows abnormally, causing the hand to deviate. Conversely, in individuals with an extra sex chromosome, such as those with Klinefelter syndrome, XXY, Jacob syndrome, XYY, or trisomy X, XXX, the shock's dosage rises to 3. The result is a predictable biological overshoot. These individuals are frequently much taller than their peers and their family members. This single gene demonstrates that the shared material on the X and Y chromosomes is not just about sex determination. It is a fundamental driver of our physical stature. This biological reality highlights that male and female are not isolated categories, but points on a complex continuum of gene dosage. This is reflected in the fact that approximately 1.7% of people are born with intersex traits, representing a significant portion of the population whose development exists along this continuum rather than within a strict chromosomal binary. Even in rare cases of sex reversal, such as XX males or individuals with Swyer syndrome, the underlying dosage of the remaining sex chromosomes continues to dictate the clinical outcome and health of the individual. In approximately 15% of individuals with Swyer syndrome who possess an XY karyotype but develop female typical characteristics, the condition is linked to specific mutations within the SRY gene that prevent the male developmental pathway from ever being triggered.
The biological impact of dosage extends far beyond height. When we examine Klinefelter syndrome, which affects about 1 in every 500 to 1,000 male births, we see a wide-ranging spectrum of effects caused by the presence of an extra X chromosome. This chromosomal addition is not biased toward one parent. The extra X is inherited from the mother in approximately 50% of cases and from the father in the other 50%. While every male has one active X, the extra X in Klinefelter syndrome is technically inactivated, but that inactivation is incomplete. About 15% of the genes on the extra X chromosome escape silencing and remain active. Many of these escapees are global regulators like ZFX, which has a functional partner on the Y chromosome called ZFY. This means that a male with XXY chromosomes has 3 active doses of these regulators instead of the normal 2. This subtle overexpression acts like a persistent background noise in the cell's communication network.
Physically, this extra dosage often manifests during puberty. Because the shared genes on the sex chromosomes influence the development of the endocrine system, the extra X chromosome can lead to primary testicular insufficiency. The testes may remain small and produce a reduced amount of testosterone, the hormone that directs male sexual development. This hormonal imbalance can lead to a variety of secondary effects, such as a decrease in muscle mass, reduced facial and body hair, and the development of breast tissue, a condition known as gynecomastia. It also impacts fertility, as the delicate process of sperm production is highly sensitive to the dosage of genes on the sex chromosomes. The subtle nature of these traits means that up to 65% of men with Klinefelter syndrome go undiagnosed throughout their lives, often only discovering their chromosomal makeup when they seek medical help for infertility in adulthood. This diagnostic challenge is sometimes compounded by the fact that Klinefelter syndrome can occur in a mosaic form. In these cases, the extra X chromosome is present in only a portion of the body's cells, while others maintain a typical 46XY count, often resulting in a more subtle physical and hormonal profile.
The dosage of shared XY genes also plays a critical role in the health of our connective tissues and metabolism. Individuals with an extra X chromosome have a significantly higher risk of developing metabolic syndrome, a cluster of conditions that includes increased belly fat, high blood pressure, and elevated levels of fats like cholesterol and triglycerides in the blood. There is also a notable impact on bone density. The same regulatory imbalance that affects height can also lead to a thinning and weakening of the bones known as osteoporosis later in life. These are not traits we traditionally associate with sex chromosomes, but they are the direct result of the sex chromosomes' role as global gene regulators. When these regulators are overrepresented, the metabolic and structural harmony of the body is pushed out of tune.
Perhaps one of the most intriguing consequences of sex chromosome dosage is seen in the immune system. Typical females with two X chromosomes generally have a more robust immune response, but are also more susceptible to autoimmune disorders. Interestingly, males with Klinefelter syndrome, who have that extra, partially active X, share this increased risk. They are more likely than typical males to develop autoimmune conditions like systemic lupus erythematosus and rheumatoid arthritis. This suggests that the dosage of certain genes on the X chromosome is a primary determinant of how the immune system distinguishes between the body's own tissues and outside invaders. Having a double or triple dose of these regulators seems to prime the immune system to be more reactive, a trait that can be protective against infections but dangerous when it turns inward.
The neurocognitive and psychological impacts of sex chromosome variations further highlight the reach of these shared genes. Both Turner syndrome and Klinefelter syndrome are often associated with specific patterns of learning and behavioral differences. In children with an extra X or Y chromosome, there is an increased risk of delays in speech and language development. Interestingly, these individuals often have better receptive language skills, the ability to understand what others are saying, than expressive language skills, which involve building a vocabulary and producing speech. There is also a higher prevalence of specific learning disabilities, particularly dyslexia and auditory processing issues. These are not general intellectual disabilities. Instead, they are specific differences in how the brain's executive functions are organized. One specific gene identified in this process is GTPBP6, located in the pseudo-autosomal region. Research has shown that IQ variability in individuals with Klinefelter syndrome is associated with the expression levels of GTPBP6. The dosage of such shared genes, which influence brain development from the earliest stages of gestation, appears to fine-tune the neural pathways responsible for communication and social interaction. We also see an increased risk for attention deficit hyperactivity disorder, though the manifestation is often different than in the general population, with individuals struggling more with attention and distractibility rather than physical hyperactivity. There is also a documented link to autism spectrum disorder, suggesting that the volume of gene expression controlled by the sex chromosomes is vital for the development of social-emotional processing. The biological consequences of dosage are even more dramatic in rarer variations, such as 48XXYY or 49XXXXY. In these cases, the thermostat is pushed even further out of range. As the number of extra sex chromosomes increases, the physical and cognitive manifestations tend to become more severe. These individuals may face more significant challenges with heart and kidney development, as well as more pronounced neurological and orthopedic issues. This dosage-dependent severity is clear evidence that the genes shared between the X and Y are not just passive passengers, they are active participants in every stage of human construction. Every extra copy of these genes adds another layer of regulatory complexity that the body must attempt to manage. While much of our understanding of dosage comes from the presence of extra chromosomes, the active maintenance of the typical state is equally vital. In females, the development and maintenance of the reproductive system is not a passive default. The FOXL2 gene, often called the pro-female gene, is responsible for actively maintaining the ovaries throughout a woman's life. Similarly, the process of X inactivation, which ensures that females do not have a double dose of most X-linked genes, is a highly regulated mechanical feat. This compaction of the inactive X chromosome is managed by the polycomb repressive complex 2, or PRC2, a molecular machine that ensures the majority of the second X remains silenced and stable.
The clinical importance of maintaining this balance persists well into adulthood, as seen in the impact of losing these shared genes during the aging process. Recent research has discovered that as men grow older, some of the cells in their bodies, particularly white blood cells, can actually lose their Y chromosome. This phenomenon is more common in men who smoke, suggesting that environmental stress can accelerate this genetic loss.
Losing the Y chromosome means losing the second copy of those vital shared regulators and pseudo-autosomal genes in those specific cells. This loss has been linked to an increased risk of developing certain cancers, such as bladder cancer and blood cancer, as well as an increased risk of heart failure and Alzheimer's disease. It appears that even in adulthood, maintaining the dosage of 2 for these shared genes is essential for preventing cells from growing uncontrollably or failing prematurely. A similar phenomenon has been observed in women as they age. Some women develop a population of white blood cells that have lost one of their 2 X chromosomes. Just like the loss of the Y in men, this loss of X-linked dosage is associated with an increased risk of health complications and a potential decline in immune function. This reinforces the idea that the shared genetic material on the sex chromosomes is a lifelong requirement. Whether it is the X or the Y that is lost, the reduction in the dosage of these global regulators leaves the cell vulnerable. The body's genomic stability is most robust when the regulators are present in their proper pairs, providing the balanced leadership that every cell needs to function correctly.
The biological consequences of gene dosage reveal that the X and Y chromosomes are far more than just sex determinants. They are the fundamental tuners of our entire physiology. From the height of our bodies to the sensitivity of our immune systems and the way our brains process language, the dosage of shared XY genes is a constant underlying force. When we look at the health impacts of sex chromosome variations, we aren't just seeing the effects of extra or missing chromosomes, we are seeing what happens when the foundational balance of the genome is disrupted. This deep integration into our biology is what makes the preservation of the shared regulatory genes so vital for the continuation of the species. As we look toward the future of medicine, the question remains, how can we harness our understanding of this dosage balance to treat the systemic health challenges that arise when the genomic thermostat is moved?
Chapter 5. Holding the Line. Preventing the Y's Decay. The human Y chromosome is often described in scientific literature as a genetic wasteland, or a shrunken vestige of its former self. But a more accurate description might be that of a fortress under a relentless multi-million-year siege. While the X chromosome has remained relatively stable and gene-rich, the Y has been stripped of nearly all its original identity, leaving behind a specialized tool for male fertility. However, this process of decay is not a simple one-way slide into oblivion. Instead, it is a high-stakes evolutionary battle, where the genome uses every trick in its arsenal to maintain the few remaining zones of partnership, the pseudo-autosomal regions, and to defend the critical boundary that prevents the total collapse of the Y chromosome. This battle is fought at the molecular level every time a cell divides, and the outcome determines the very future of our species' genetic architecture. To resolve this, researchers utilized single-cell sequencing of human sperm and compared the DNA of humans to 7 other primate species, including chimpanzees, gorillas, and macaques. By constructing an evolutionary tree of these sequences, they discovered that the pseudo-autosomal boundary has been remarkably stable, remaining unchanged for roughly 25 million years, since the common ancestor of humans and Old World monkeys. The most recent data confirm that this boundary is not a fuzzy zone, but a precise molecular wall, narrowed down to a window of just 201 base pairs. This stability is a testament to the intense selective pressure to keep the sex-determining region, particularly the SRY gene, which sits a mere 10,000 base pairs away from this boundary, locked away from the high recombination environment of the pseudo-autosomal region. If the SRY gene were to cross this boundary and enter the zone of frequent genetic swapping, it would be transferred to the X chromosome with catastrophic regularity, leading to widespread sex reversal and infertility.
The primary reason this boundary is so fiercely defended is the threat of Muller's ratchet. This is the evolutionary process where a non-recombining chromosome, like the Y, inevitably accumulates harmful mutations over time.
Because the Y chromosome cannot swap DNA with a healthy partner to edit out these errors, the overall quality of the chromosome can only decline. It is like a digital file that is copied over and over again without any error correction software. Eventually, the data becomes corrupted. To counteract this, the Y chromosome has evolved unique structural defenses. One of the most fascinating is the use of massive palindromes, long sequences of DNA that read the same forward and backward. These palindromes allow the Y chromosome to engage in gene conversion, a process where it uses one side of the palindrome as a template to repair a mutation on the other side. It is a form of self-recombination that doesn't require an X chromosome, and it is the Y's primary defense against total genetic decay.
However, the battle to maintain the Y chromosome is not just about preventing loss, it is also about the surprising ways the chromosome can expand. We often assume that the Y only gets smaller over time, but research into other species shows a much more dynamic picture. In the plant world, specifically in the species Silene latifolia, the Y chromosome is actually significantly larger than the X. This expansion is driven by the accumulation of transposable elements, jumping genes that copy and paste themselves throughout the genome. One specific family of these elements, known as Athela, is heavily overrepresented on the Y chromosome of these plants. These repetitive sequences act like genomic filler, puffing up the size of the chromosome even as its functional gene content might be shrinking. This suggests that the life cycle of a sex-limited chromosome might involve a phase of massive expansion before it eventually contracts.
However, the battle to maintain the Y chromosome is not just about preventing loss. It is also about the surprising ways the chromosome can expand. We often assume that the Y only gets smaller over time, but research into other species shows a much more dynamic picture. In the plant world, specifically in the species Silene latifolia, the Y chromosome is actually significantly larger than the X. This expansion is driven by the accumulation of transposable elements, jumping genes that copy and paste themselves throughout the genome. One specific family of these elements, known as Athela, is heavily overrepresented on the Y chromosome of these plants. These repetitive sequences act like genomic filler, puffing up the size of the chromosome even as its functional gene content might be shrinking. This suggests that the life cycle of a sex-limited chromosome might involve a phase of massive expansion before it eventually contracts.
The existence of the second pseudo-autosomal region, PAR2, presents its own evolutionary paradox. While PAR1 is found in most mammals and is essential for male meiosis, PAR2 is a much more recent addition, appearing only after the human lineage split from chimpanzees. It formed when a piece of the X chromosome was duplicated and moved on to the Y. What makes PAR2 so strange is its low recombination rate. Unlike the obligatory crossover in PAR1, recombination in PAR2 occurs in only about 1% of male meioses. This means that for most of its history, the DNA in PAR2 is inherited just like a sex-linked region, staying on the X or the Y for generations without swapping. This low activity allows the X and Y versions of PAR2 genes to slowly drift apart, creating a genetic signature that is halfway between an autosome and a sex chromosome. Some researchers believe PAR2 might be a pseudo-autosomal region in the making, or perhaps a redundant backup that hasn't yet been fully integrated into the meiotic machinery.
This constant shuffling and shifting of the sex chromosomes is part of a broader phenomenon known as sex chromosome turnover. In many lineages, particularly in fish and amphibians, the identity of the sex chromosomes can change with startling speed. A classic experiment involving swordtail fish documented a 30-year period where a sex-determining region translocated from one chromosome to an entirely different autosome, effectively creating a brand new sex chromosome pair in just a few decades. In some cases, the battle for recombination is bypassed entirely.
Certain species, such as specific voles and flies, have evolved sex chromosomes that do not recombine at all because they undergo achiasmate meiosis, a specialized form of cell division that ensures proper segregation without the need for genetic swapping. This turnover prevents the total collapse of the reproductive system by starting the clock over with a fresh, healthy pair of chromosomes.
The possibility of the human Y chromosome eventually disappearing altogether is a subject of intense debate among geneticists. Some point to species like the mole vole or certain Japanese spiny rats which have lost their Y chromosome entirely. In these animals, the master sex-determining switch has moved to an autosome, and both males and females have an XO chromosomal makeup. Proponents of the disappearing Y theory argue that at the current rate of gene loss, the human Y could be gone in as little as 10 million years. However, others argue that the Y has reached a state of evolutionary stasis. They point out that the genes remaining on the Y today, particularly those involved in global gene regulation and sperm production, are so essential that they are protected by intense purifying selection. The Y may be small, but it is now composed of a hard core of indispensable genetic material that is unlikely to be lost.
The possibility of the human Y chromosome eventually disappearing altogether is a subject of intense debate among geneticists. Some point to species like the mole vole or certain Japanese spiny rats which have lost their Y chromosome entirely. In these animals, the master sex-determining switch has moved to an autosome, and both males and females have an XO chromosomal makeup. Proponents of the disappearing Y theory argue that at the current rate of gene loss, the human Y could be gone in as little as 10 million years. However, others argue that the Y has reached a state of evolutionary stasis. They point out that the genes remaining on the Y today, particularly those involved in global gene regulation and sperm production, are so essential that they are protected by intense purifying selection. The Y may be small, but it is now composed of a hard core of indispensable genetic material that is unlikely to be lost.
The battle to maintain the Y chromosome also involves the evolution of dosage compensation. As the Y decays, the body must find ways to balance the expression of X-linked genes. While we often focus on the somatic system of X inactivation, there is also a critical need for germline-specific dosage compensation. In the cells that produce sperm and eggs, the regulatory environment is entirely different. Misregulation of gene expression in these cells is a primary cause of hybrid sterility and inviability, a pattern known as Haldane's rule. This phenomenon is often linked to the large X effect, which describes how the X chromosome plays a disproportionately significant role in reproductive isolation. Because the X is so central to the regulatory health of hybrids, any mismatch in these shared XY systems can lead to the sterility or inviability of the offspring. When two different species interbreed, the conductors of the genomic orchestra, those shared XY regulators like ZFX and ZFY, may not be able to properly manage the foreign DNA of the other species. This leads to a breakdown in gene expression that is most severe in the heterogametous sex, the XY male in mammals. This highlights that the shared genes on the X and Y are not just internal managers. They are the guardians of species boundaries.
The preservation of the shared regions is also seen in the way the X and Y chromosomes utilize their physical environment during meiosis. In the early stages of sperm production, the sex chromosomes are sequestered into a specialized compartment called the XY body. This compartment is a hub of intense epigenetic activity, where the chromosomes are chemically modified to suppress most of their gene expression, a process called meiotic sex chromosome inactivation. However, the pseudo-autosomal regions must remain partially exempt from this silencing to allow the recombination machinery to do its work. This creates a delicate balance where most of the sex chromosomes are turned off, while the tiny shared tips remain turned on.
This spatial and temporal regulation is a masterpiece of cellular coordination, ensuring that the chromosomes can pair up without interfering with the rest of the cell's functions. The evolutionary dynamics of the pseudo-autosomal regions also reveal a surprising amount of genetic diversity. Because these regions recombine so frequently in males, they are hotspots for the creation of new genetic variants. This diversity can be a source of evolutionary innovation, but it also means that the pars are more susceptible to certain types of mutations than the rest of the genome. For example, the high rate of DNA breakage and repair required for recombination can sometimes go wrong, leading to deletions or duplications that are associated with developmental disorders. The genome must balance the need for meiotic pairing with the risk of creating these copy number variations. This balance is another front in the evolutionary battle, where the benefits of a functional Y chromosome are weighed against the risks of a high recombination environment. Comparing the human system to that of the platypus offers a glimpse into a very different strategy for maintaining sex chromosomes. The platypus meiotic chain, with its 10 chromosomes and 8 pseudo-autosomal regions, is a vastly more complex docking system than our own. This chain must be maintained with absolute precision. If any one of the 8 links fails to recombine, the entire chain can break, leading to catastrophic aneuploidy. This suggests that the battle to maintain homology can take many forms. In humans, we have simplified the system down to 2 small regions, while the platypus has doubled down on a complex multi-link chain. Both strategies have been successful for over 100 million years, proving that there is no single right way to build a sex chromosome system. The struggle to prevent the total decay of the Y chromosome is also reflected in the evolution of sex-determining genes themselves. While SRY is the master switch in most mammals, other lineages have co-opted different genes for the same role. In birds, the DMRT1 gene is the primary driver of sex determination, and it is located on the Z chromosome. In some fish, the sex-determining locus can move from one chromosome to another, a process that can be influenced by environmental factors like temperature. This flexibility shows that the male-determining function is not tied to a specific piece of DNA, but is a role that can be played by many different genes. The Y chromosome is simply the current vessel for that role in humans, and its survival depends on its ability to maintain the essential shared genes that allow it to function within the broader genomic network.
Comparing the human system to that of the platypus offers a glimpse into a very different strategy for maintaining sex chromosomes. The platypus meiotic chain, with its 10 chromosomes and 8 pseudo-autosomal regions, is a vastly more complex docking system than our own. This chain must be maintained with absolute precision. If any one of the 8 links fails to recombine, the entire chain can break, leading to catastrophic aneuploidy. This suggests that the battle to maintain homology can take many forms. In humans, we have simplified the system down to 2 small regions, while the platypus has doubled down on a complex multi-link chain. Both strategies have been successful for over 100 million years, proving that there is no single right way to build a sex chromosome system. The struggle to prevent the total decay of the Y chromosome is also reflected in the evolution of sex-determining genes themselves. While SRY is the master switch in most mammals, other lineages have co-opted different genes for the same role. In birds, the DMRT1 gene is the primary driver of sex determination, and it is located on the Z chromosome. In some fish, the sex-determining locus can move from one chromosome to another, a process that can be influenced by environmental factors like temperature. This flexibility shows that the male-determining function is not tied to a specific piece of DNA, but is a role that can be played by many different genes. The Y chromosome is simply the current vessel for that role in humans, and its survival depends on its ability to maintain the essential shared genes that allow it to function within the broader genomic network.
The X and Y chromosomes are far more than a biological mismatch. They are a testament to evolutionary resilience. By retaining shared pseudo-autosomal regions and vital regulatory gametologues, these chromosomes ensure the precise mechanics of inheritance and the balanced orchestration of our entire genome. This shared heritage dictates everything from our physical stature and immune responses to how our brains develop. The persistence of these genes reveals that the Y chromosome is not merely a decaying vestige, but a specialized conductor essential for life. As we map the sexome, we find that our biological identity is defined not just by our differences, but by the ancient threads that still bind these two chromosomes together. Understanding this balance is the key to unlocking the future of personalized medicine. Thank you for listening to this exploration of our genetic past and future.